Parasitology. I began working with the site about the time that the Manter Lab got very deep in the DAMA protocol and the Stockholm paradigm. And we began thinking about coprolites and archaeological residues as essentially deep time components of DAMA. So essentially, as I've said here, coprolites are desiccated archives of organisms that lived in or passed through the gut. So that contributes to D, the document in the DAMA protocol. Within the past, 15 years, we become aware of the fact that uh, the La Cueva de los Muertos Chiquitos, I'm just going to call it Zape from now because it's located in El Rio Zape in Durango, Mexico. So that the Zape coprolites are not only human coprolites, but they're canid coprolites. And so I'm waiting with bated breath for my molecular colleagues to provide us with some more information about that. So they can provide us with human companion animal interaction insights as to what parasites were passing between humans and domestic animals and what wasn't passing through. So the talk outline is a little complicated. We're going to look at La Cueva de los Muertos Chiquitos. We're going to look at Zape, and we're going to also look at the ancient environment. Now, to demonstrate how valuable coprolites are as a desiccated archive of organisms, I'm going to go through the gut microbiome analyses of the past few years first. Then to explore contamination and how parasites distribute themselves in sediments, I'm going to go through the sediment analysis that was the focus of the latest dissertation by Elisa Puca de Orujo in Brazil. And then we're going to get to the coprolites that date later than the sediments. Important to me is that Zape, represented by a star, is on the very northern frontiers of Mesoamerica. When we get into the southern reaches of Mesoamerica, the preservation potential gets worse and worse and worse. Therefore, having a sample from Mesoamerica provides us our first glimpse of what could be the diversity of parasites as we get to the more complex, stratified, and elaborate cultures of Mexico. The vertical profile of the cave looks like this. So there's a Rio Zape down below, 50-foot slope, and then there's a trail that accessed the base of the cave. To get to the cave, the prehistoric people carved steps, well, actually depressions in the rock, handholds and toe holds to climb into the cave. The archaeologists selected this cave among all others to excavate because of those toe holds. They reasoned that it would be impossible for animals to get into this site. Only humans could get there. So when we discovered canid coprolites or canid parasites, it was a bit of a shock because the site had been selected for human access only. So that's interesting to keep track of. The upper ledge is not an issue for us. We're going to be talking about primarily the remains that were found in the back part of the cave and then deeper levels in the back. Just to point out that the entry is here. So this is a plan view, and this is a grid outline of the excavations. The excavations were done in 1957, 1959, and 1960. They were done in 6-inch levels and 5-foot grid squares, which is enormous compared to modern excavations. By the 70s, the excavations were being done in 10 centimeter units and grid single grid squares. So if you're an archaeologist and you look at this, you immediately think, well, that's a tiny site because you're thinking in terms of meter grids. So keep in mind, it's 25 feet from this edge of the trench to the wall. So it's a fairly large site. That would be about 75 meters, I do believe. So it's a large cave. So 5 by 5 grids. And the first excavations were focused on a room, which was discovered in the back, which also had a raised platform. So a room and a raised platform, which is suggestive of some sort of elaborate, maybe perhaps ceremonial activity. Today, the region looks like this. This is a photograph of the topography near a town called Zapechico. And it ranges from semi-arid and cultivatable to arid environments such as this. And these are the cave landforms. So La Cueva de los Chiquitos Muertos is consistent with the caves that we're seeing here. If we look at 1960, this photograph was taken looking out of the main cave entrance towards the Zapechico, and there's several different things that strike me. First of all, this stream is entrenched now, and it looks to me like the water level has gone down, and that prior to this, there was a broader, a broader floodplain, which would be been arable in prehistoric times. And the fact that it was arable is indicated by there's a special variety of beans that is found in this archaeological zone. So there's a Zape bean variety that you can buy now and grow in your backyard if you would like. Now, something that was really interesting that came through last year from Lawrence David's molecular genetics lab at Duke University was a series of sequences from the coprolites that represent animals that were eaten at the site. What struck me was the fish, because some of these I recognize as endangered desert fish for Mexico and the southern New Mexico and Arizona and Texas as well in the United States. So we have been in southern Arizona struggling to restore uh, environments specifically for stone rollers and chubs. That would be Campostoma and Gila conspersa. So let's look, based on the fact that we know what the environments for these fish were, what could the environment look like for Rio Zape at the time that the cave was occupied? Gila conspersa in Arizona is associated with cienega environments and slow-moving shallow streams. It's a chub, a cute little fish. Now, the stone rollers in Arizona, again, are associated with the reconstituted environment of the Gila River in the Mojave region at what's called the Great Bend of the Gila River. So this is the environment we would expect for a Campostoma. And for the suckerfish, which are rather large, they are found in the Rio Yaqui today, which I believe is in Sonora and Sinaloa. And so these are the kinds of environments that these fish would have been associated with in prehistoric times when the people were using the Zape cave. So in looking at the environment now, as it is entrenched and desertified, I think that in prehistory, it would have looked much more like the Gila Bend at the Gila River. So thanks to Lawrence David's data, I can now see this environment in my mind when I look at the setting of the Rio Zape today. So we're going to look at microbiome studies first because coprolites are an archive of ancient bacteria. The latest molecular data includes perhaps 100 sequences from various bacteria and protozoa, and I've shared some of those with Scott and some of the other Manter folks. But I think we're on the verge of some very, very exciting parasitology through molecular biology beyond what I'm going to share with you today. So let's talk about the first papers focusing on the microbiome of Zape. This is a pretty cool one. It's Raul Tito is a senior author, but Cecil M. Lewis, the final author, is the individual that was corresponding with me over the time. I think Cecil is now taking over um, uh, a program office job position at NIH. So this first paper showed that natural selection influenced the microbiome functional profiles. Why is that important? It's important because as we look back now, after conducting several of these studies, the functional profile of prehistoric folks was most influenced by the high fiber diet and the types of fiber that was in those diets across the Americas. And therefore, human microbiomes were structured geographically during pre-Columbian times based on what types of foods were available that would contribute to that gut microbiome. For this site, Elisa Pucco del Rujo and I went through the prehistoric foods at Zape to relate them to the Tarahumara, which would be the geographic connection between Zape and a modern descendant group. Subsequent to that, further studies discovered kind of something interesting to me, that as one sample had a taxonomic representation of a weaning child because of the milk representation in the diet. Why is that cool? One of the first studies we published based on work we did in the 1990s was to review the demographics of the site by looking at dental casts in wads of fiber that was spit out from feasting in prehistory. We found casts for 49 different individuals at this site, indicating that at least for ceremonial purposes, feasting on agave, a lot of people would be represented there. We found dental casts from children whose molars had not fully erupted, to old people who had abraded and lost all of their teeth due to abscessing. So what's interesting to me is a child microbiome is another bit of data showing that communities, including children, were involved in this and at this site. The other thing is that this study found that the Zape the DNA was preserved better than any other part that had been examined in the world at that point. And that included mummies from Chile and coprolites from Texas. Weibo was a senior author of a very time-consuming study. The end results of this were we could find DNA from only 3 sites. I did a survey of 77 coprolites from 10 sites, and we found that only 7 of those coprolites were suitably preserved for DNA recovery. That lesson led me to publish a paper on taphonomy and decomposition of coprolites, so that other folks could screen coprolites relatively rapidly uh, compared to the year I spent sorting the coprolites for analysis. Now, what the data showed is that 39% of the prehistoric sequences were undescribed, and that suggests we've lost a lot of bacteria over the years, over the centuries. And compared to present day gut microbiomes, coprolites are more similar to non-industrialized people. So archaeological samples show a lower abundance of antibiotic resistant genes and mucin degrading genes. What intrigued me is that a site in the study that was reliant on grasshoppers, there was increased chitin digestion genes found in that sample. So this is a very important sample. And again, it shows the value of coprolites as archives for gut organisms. Now, Cecil Lewis, who I mentioned as being in those +1 articles, let me know about 2002 or 2003 that pinworm eggs were turning up in coprolites with a canid gut microfossil or gut microbiome um, content. So that brought to mind the potential of contamination. And could we find that? Well, there were two studies that contributed to this. This is the Copro ID approach to identify what animal deposited the coprolites. And the Copro ID relies on two factors, host DNA and then species microbiome. The microbiome is called source predict. So essentially for each sample, the microbiome was characterized as being either canid or human. And the host DNA was then identified as human or canid. Now, throughout this article, I think we were assuming that dogs were represented, so that source predict was based on dog DNA or dog gut microbiomes. And we have to admit that gut microbiomes of dogs today are different from what they were 600 years, 800 years ago when this site was being used. And that the gut microbiome of humans and dogs may have been quite similar in prehistoric times, since dogs were eating human feces and both animals were eating essentially the same sources. So this was the interesting aspect of this study. In red on the left, we see the data for human source predict and DNA from the 8 coprolites they studied. On the left, the canid host DNA and source predict. What was interesting is that for samples 2 and 3 and, 2 and 23 and 29, the host DNA was canid. However, the gut microbiome was human. And this led to the question, why, of course, and part of the answer could be cross-contamination. Another one could be that humans were eating dogs and then therefore were passing canid DNA. The third is that these two host species were eating each other's feces. And it's difficult for us to imagine how this would happen in our modern environment. But I'm sure it happened in medieval environments where humans were sharing their houses with canids, or even in caves. And I would imagine even in historic Arctic villages, there could have been a pretty significant cross-contamination of humans and canids by eating each other's feces in a dirty environment in which people are essentially interacting with their companion animals in trash. Now, all of those samples that were analyzed by the copper ID method had been analyzed by the archaeoparasitology class, and the data were published by Agustin Jimenez. And we see that the microbiome data is represented, and all of the microbiome copper ID or source predict were human. Host DNA was variable with canid DNA and human DNA. However, what we published from this was that we found Enterobius vermicularis predominantly in the sites. And then the coprolites that are represented by asterisks, they were between 1,000 and 2,000, 5,000 eggs per gram, which indicates that that was patent infection of pinworms. So I think that the parasite data support a human infection for sample 2. Trichuris trichiura, a human infection for sample 5. Dipallidium is probably a dog because of the canid host DNA and dipallidium in the gut. However, 25 and 27 both have an abundance of DNA and are human. In sample 29, I don't know what to make of this. Host DNA is canid, even though the gut microbiome is consistent with human, but there is essentially one egg of Enterobius vermicularis that was found in this site. And I'm beginning to consider the potential that we have egg movement in the post-deposition environment from one coprolite to another. So from animal coprolites to human coprolites and vice versa. The other thing that is important is that this ancylostomid, which we identified as most consistent with Ancylostomid duodenale. Lauren Cleland sent me an email that she had sequenced Ancylostoma caninum from this sample. So we could easily be dealing with a dog. So I put a question, I mean, with a canid. So I put a question mark there. Now, to explore more fully whether there are feces being ingested, Richard Hagen led this group of researchers looking at the signals of canid and human microbiomes. In actuality, this paper was a comparison of extraction methods for recovering ancient microbial DNA. But what was fortuitously discovered is that every analysis with all the different methods for sample 2 showed dog feces. So the arrow is pointing to that dog fecal microbiome signature in sample 2. Now, sample 2 has an abundance of pinworm eggs. It is human DNA, and I think this signals that humans were somehow ingesting animal or canid DNA in the prehistoric filthy environment of the cave. Now, I'm going to go and explore more the potential of cross-contamination in the sediment analysis. And when Cecil Lewis told us about this potential of contamination around 2005, Jonica and I began exploring the sediment analysis, and in a brief examination, we found that there were pinworm eggs in the sediment. So that led to the potential that we had egg contamination of materials in the site. Okay, checking time. So this is a history of the excavations. In 1957, 2 parallel trenches were dug, and those are at the back feet of the backside of the the cave, 5 feet wide and 23 inches long. They excavated this and an additional 2.5 foot trench to expose a house floor and define the walls of that house floor, and a platform built in the back of the cave. 1959 and 1960, additional excavations were carried out in the cave for another 15 feet towards the front of the cave. Now, in grids D1 and D2, C0 and C1, burials were found beneath the coprolite deposits and were capped by an adobe floor. So essentially, burials in that part of the cave were the first thing that was done at the site. It was a burial site initially. The burials were capped with an adobe floor. Then trash at some point, and that was between 700 AD and 850 AD, trash began to be deposited on the burial floor, the burial cover, excuse me. So we have a separation of the sediments, which is important to keep in mind, from the coprolites by a sealed adobe level. So let's take a look. So this is 1959. The excavations are focused on the house. And so that is a mistake in my chart. The adobe floor over the burials was actually at these grids. So we will go to here. So this was excavated in 1959. That is the house floor in the elevated area. This area was excavated in 1960, and this is the area of the floor over the burials. So the site is named La Cueva de los Chiquitos Muertos, or Muertos Chiquitos, because the burials were all of little children. So that's the cute kids, I think, is how it would be translated, who were dead, underneath that floor. So does everybody follow me? In this area, excavated originally because of a room in an elevated platform, this is the cultural deposits that attracted the archaeologists. Later, they excavated this area, found a midden, and beneath that midden, they found a floor which covered a series of burials of children, and those children died more or less simultaneously.
Wow. Yeah, we're following you. That was a good explanation.
Thanks. I think it was Elise and I weeks to decipher the field notes. Uh, Jonica did an initial analysis of human specimens. Then she was able to find human-specific and canid-specific parasites in the same samples. Now, I want to bring up golden spider beetles because these are the agents of decomposition at this site, and they are ubiquitous in the middens, in the coprolites. And the golden spider beetles decompose dry organic remains, so they moved in to eat the feces after they had dried. And then they burrow into the feces. I'll show you pictures of this, it's beautiful. And then hollow them out. Then the feces collapse, the coprolites collapse, and then eggs probably spread in the environment and around to other samples. So then we went into the burials to see if golden spider beetles were ubiquitous. Want to go to show you the typical coprolites that are well preserved. You see in a lower images are diarrheal samples, and the upper images are form samples. But there's a golden spider beetle, and this is what happens to a coprolite that's been infested by the beetles. This is a single coprolite, outside view and inside view, and you can see the holes that are burrowed in, and then the interior is eaten out. So a fascinating a fascinating aspect of decomposition for the caves. So, did we find golden spider beetles in the burial levels? So under the adobe floor, there were 7 samples. They were composed of trash mixed with non-cultural sediment and geological sediment. In some cases, the samples were taken near the head of the interments. So Elise and I went through all of the bags, the field collection notes around to the left, and then we analyzed the material to figure out whether it was organic material that might come from a burial, or trash that might have been thrown in with the burials, or just geological debris that it was a fill for the burials. So here we have sample 1, soil from a burial. What we found was a mixture of trash, what archaeological term is midden, and geological debris. The limestone sediments that we found were generally larger than a millimeter, indicating that it was from dust that had settled from the top of the cave. So then trash is indicated by lizard scale, grass, seeds. There was human hair, and then cotton threads. Soil close to the scalp, which is sample 3, was geological and mid material, limestone. There was string and cloth and leaves. So that's mostly trash and geological sediments. Sample 4, soil at the pillow and scalp. So they had interesting pillows that were made of, they're essentially a platform like you'd find in I associate these with Asian styles of sleeping. But there are burial wrappings. Importantly, the trash of nut fragments, agave leaf bases, were mixed with cloth and agave matting that would have come from the clothing and wrapping of the burial. So sample 4 looks like a burial. Sample 5, geological refuse with burial wrappings, and specifically agave fragments, but there are also seeds from trash. Then sample 6, we screened this to hopefully make it easier for analysis, but we found geological fill and burial wrappings. In sample 7, the same geological fill with burial wrappings. So only sample 4 showed a pretty good signal of a sample that actually came from a burial. When we look at the insects, the golden spider beetles are ubiquitous, but look, they're also dermestids and also decomposer flies, which indicate that the graves were open long enough for uh, for flies to do their life cycle there, and then to then dry out enough for domestids to move in and do their decomposition. Pretty fascinating stuff. So what do we find with regard to parasites? Wow. We had to say wow twice because in the previous 2 decades of analyzing the burials, we'd never seen Ascaris lumbricoides, we'd never seen Toxicara, we'd only seen Trichuris a couple of times. Yet in these low sediments underneath the coprolites, there seemed to be a different spectrum of parasites, which is interesting. It suggests to me that there are 2 occupations of this site that are represented only by the parasites. Then with regard to sample 4, which is our sample that is most likely from a burial, we find lots of pinworm eggs, which suggests that the individual may have been infected at the time of burial. The other samples that were a mix of trash and sediment also have parasite eggs, pinworm eggs, which suggests that the pinworm eggs were distributed throughout the sediment from whatever their source was. We also find Physoloptera eggs, which indicates that canid parasites were distributed as well. All right, let's go above the burial cover and look at what do we find in coprolites. let's see how my time is going. Well, the first paper by Augustine and Martin Fogaça from Brazil participated in this. Um, what we found was an echinostoma, something related to it. I think we've decided that it's a fluke in later analyses. We found hymenolepids and we found Dipylidium caninum. Uh, and we found a lot of pinworm. We found ancylostoma and we found trichuris in 2 samples. So this was a spectrum that we were working with when we were beginning our analysis. This is out of 34 samples, which I didn't put in there. Augustine and I selected samples that looked to be the most human based on the copper ID, which was done on 8 of these samples. We know that one of the samples is likely from a dog and potentially a second one is from a dog. Or excuse me, a canid. I don't know if they're dogs yet or not. Now, Jonica processed 100 coprolites and recovered parasites from these samples. Last year, I went through all of the samples to determine which are human and which are dogs, in which I could not identify. 3 samples I could not identify as to have a human origin or a dog origin. So I did not consider those in subsequent analyses. But when we look at that big sample, we find that of the 26 canid coprolites, 37 of them have Toxascaris, 8 have percent, 8% have Physoloptera, 3%, 13% have the trematode, and we did not find Taeniodin in this series of coprolites. The human samples, there was 1 Taeniod, which was represented by a single egg, probably a contaminant. Then 71, 6% had Toxascaris, 1% had Physoloptera, and 1% had trematodes. So I think what this is telling me is if we have cross-contamination, we do have samples of that we have eggs from animal samples that are being incorporated into human samples. And if we look at the pinworm data, it's going the other way. So 8% of the canid samples have Enterobius vermicularis, and 47% of the human samples, 71 of which have Enterobius vermicularis. So I think in looking at these data somewhat objectively, I think we have cross-contamination. I think those little golden spider beetles are decomposing samples, and the eggs are being introduced from the sediment in the holes that they make in what look to be intact samples. What that means is that we have to take our overdispersion data and look at them with a more conservative mindset when we get to redoing reevaluating those data. With regard to ELISA work, Jonica analyzed the samples for Cryptosporidium, and 19 canid samples, 58% of them were positive, 68 human samples, and 74% of those are positive. So in summary, when we look at our data, we can see that from the burial sediments, we have Trichuris trichiura, Toxocara canis, Ascaris lumbricoides, Anaerobius, of course, and Physoloptera. From the canid coprolites, we have that series of parasites, which I've talked about already and delineated. And then we have some human-specific parasites among the humans, as well as ancylosimids. And I think a case can be made for Ancylosima duodenale, but it's circumstantial, and I won't go into that today. And then we have Cryptosporidium parvum. Now, the molecular approach to pathogen DNA has begun with Joe Brown at the Gilling School of Global Public Health and the University of North Carolina. So these are his preliminary data of multiplex qPCR assays. And what is quite exciting for me is we're looking beyond what we can see with a microscope, and what we were able to recover through ELISA. So we're seeing blastocystis, a lot of Escherichia coli of various toxic varieties. We're seeing Shigella, we're seeing Giardia, and of course, pinworm, which is the most prolific parasite among the people at Zape. And this is really exciting to me because every decade we turn a new chapter, it seems, since we started looking at these in the 1990s. And what I can see here is a premonition of what we're going to be able to recover with bacteria and protozoa in the not too distant future. So right now, Joe Brown and Lawrence David are refining methods to detail even more about host feeding, what humans are eating and their dogs. The host source, are we looking at canids in the terms of dogs or a different canid? And pathogens, including vector-borne protozoa. Very, very excited about that. So I think I have to thank new colleagues and emerging methods. And I think that the Zape has become such a famous site for its extremely well-preserved DNA relative to other sites, that people are attracted to this. I find that I'm transitioning from a research role to a caretaking role with regard to the samples. So combined with the data are increasingly serve as a document of ancient endemics and the changing of environment of Durango, Mexico. For this reason, they fit into the DAMA protocol of documenting what parasites existed in a primeval environment um, before modernization. We'll take a look at some parasites. Um, Here is Martin Fugasa in Argentina, found this parasite, and I love this because the hooklets are preserved in the oncospheres. It's a beautiful, beautiful image. This one is Ancylosum caninum, and Augustin Jimenez took this picture in the Manter lab, and you can clearly see the little mummified juvenile within the egg. And then we have pinworm, again, those beautifully preserved juveniles inside the eggs. What I love to do with a microscope is put pressure on the eggs, and sometimes I'm able to squeeze a larva through the opercula, and then I laugh. Okay, thank you very, very much for sitting through this, and I hope that it was useful.
Thanks, Carl. I have a question.
I have an answer, maybe.
Okay. What kind of evidence is there for the relationship between humans and whatever canids might have been there? For example, did these folks have quote pet dogs or hang-on dogs or hang-around dogs? I can't imagine that coyotes would be so fearless as to be that in that close of association, but what do we know about humans and their dogs?
Well, I hope I'm not speaking out of turn, but Lawrence Davids has found Canis DNA in all the samples. So we know that they're associating with Canis, but it's not dog. He's finding Canis vulpes. Now, does that make sense? Yeah. We're at the northern edge of Mesoamerica. And in uh, especially central Mexico, it down into the margins of the Mayan area. There's an important symbolic value put on wolves. So they're associated with one of the gods of war and rain. And also, there's a hybrid of dog and wolf that was especially important religiously, because this hybrid carried the blood and the spiritual value of the wolf, but was manageable because it was the behavior is more pliant because it was domesticated with a dog. Now, when we had no idea there were canids at this site until the parasites turned up. And I think it's very telling that the Echinococcus scolix is rotating at this point, probably in excitement. But I had wondered why there were these canids at the site. However, the elevated platform mound and the house in the back was unusual to me. The elevated platform kind of signals a ceremonial use. So my hunch is that they were getting Mexican wolf pups or Mexican wolves and then bringing them into the cave and domesticating them well. And then I think that was a source for ceremonial use in the area, but potentially these were then traded to other parts of Mesoamerica. So I'm very excited about that finding. However, tomorrow they're meeting with the team that has done more refined sequencing to determine whether or not I can say that formally. I think from their analyses, we're seeing some fantastic data that will have to come out in several different papers.
See, of all of the years I've heard you talk, Karl, I really have not heard very much about canids, and and it's really, you know, today your talk sort of stimulated some questions about that. Wolves, and from watching, for example, Brent Nickel try to domesticate a coyote pup at Cedar Point, I can tell you that this is a non-trivial task. And wolves for sure, and wolf dog hybrids, I cannot believe that those are lap dog puppies. This is not a this is not a trivial matter. So the relationships with canids would be kind of an interesting, for me at least, to hear. So,
The other thing is the whole idea of bringing them to a cave that's
yeah.
inaccessible is not from the perspective of keeping animals out, it's to keep them from getting out. So I think it's one aspect of of bringing them together, which also explains why human feces and canid feces were mixed together in this activity area of filth. And this is only 5 feet away from the feast deposits. So those agave quids are deposited primarily in the northern part of that cave, in the room and the platform. So that was where as many as 49 people were coming together to feast at one time. And from my perspective, having a feast that close to feces is not, is kind of repulsive to me. But perhaps it was not to our ancestors.
Thanks.
Any other questions?
Hey, Carl, I mean, I always heard, that sources of protein, right, and so forth were not I mean, not like today, right? Where we're not as protein. I mean, the protein limitation was an issue in those parts of Mesoamerica. I mean, is there any evidence perhaps that dogs were also supplementing the diets of humans?
Yes, and we found that in the two sites it's been discovered. About 208 Common Era, 200 AD. There's a site in southern Utah where a PCR analysis recovered dog DNA. We did a similar analysis dating to 1200 from Canyon de Chelly in Arizona using protein residue analysis. And among the different animals ranging from turkeys to bighorn sheep, there were dogs as well there. And certainly with regard to the CoPro-ID, the explanation that they were eating dogs or canids could fit into this. The other thing that we have to consider is that there were no dog bones found at this site, no animal, no canid bones. I need to drive to the archive in Las Vegas and go through the remains and see if that is actually the case. But the things that Mesoamericans used dogs for were not only food, wolves for, and wolf hybrids, not only food, but also to make paraphernalia for masks. They had a method of self-sacrifice by bloodletting with sharp owls that would press into their genitals or their ears or their ankles. So, and those are made of wolf or puma bones. Valeria, did you have a question? Are you still here? I think she left. So Valeria had a question, but I don't know what it was.
Looked like she had to take off.
Okay. Anything else?
A question by Valeria is available in the chat. It
Oh,
was
there's
about
the
the
chat.
insects.
Okay. Chat. Okay, I have to turn this off. Chat. Okay, the question is, do we look for presence of potential vectors of parasites? No. And part of that is I look for I look for lice, I look for fleas, I look for ticks, and people will eat those and and ingest what might have been ingesting them. I think that's a strategy. I think so. I have had some success at finding ticks and some success at finding lice, but I've never been able to find a vector. So I have developed a strategy for looking for triatamines, but that hasn't borne fruit yet. Okay, did that answer the question?
Yeah, seems so. I was wondering about the triatoma also down there, but I know you hadn't seen any yet.
I have been told that there are bags of sediment from the site, and I think it'd be very, very worthwhile doing a 1/8-inch screening of those sediments to see if we can get bits of insect and identify some triatamines. I think that area, the cave, the environment is ideal for the cycling of Chagas disease.
Yeah, it
So
seems like it. Have you been there?
No,
No, because it
I've
would be
wondered-
interesting to see if there's any wood rats around there that have some middens that they've developed.
well, I'm hoping that the DNA has showed two species of Paramiscus,
Oh, yeah.
But we will wait to see if we can get DNA. The site itself became in the control of a drug cartel in the 90s.
Ah,
And up until the time that Augustine was here, it was still in control. However, I have seen that it's turned up on Mexican tourist sites, even tours of archaeological sites in Rio Zape with
wow.
pictures of the caves. So perhaps if Augustine or you will go down with me, I want to go with, or Roberto, I want to go with a Spanish speaking person. Because I don't think my leftover Portuguese will get me very far.
Ah, well, it might get you too far, actually.
Yeah.
Okay, well, any other questions? Thanks, Carl. Anything, I don't see anything else here on the text, so, and I've recorded this, so I will let the thing download and then do its thing, and I will then post it on the Facebook. Unfortunately, I didn't get Facebook announced earlier because there was some problem with Facebook the other day when I tried it, but now it's working, so. Carl, that was amazing. Really
Thanks,
cool.
Carl. Thanks,
Really
You
cool.
Carl.
guys are welcome.
Okay, so remember,
Thanks,
no
Carl.
seminar, no seminar next week, and we need to have someone give someone volunteer for the next time. Thanks, everybody. Thanks, Beth.
Bye.
Bye, everybody.
You guys take it easy.
So Scott, I can do the next one.
All right, well, we do have the next one set up already, so John McKenzie will give the next talk in 2 weeks.
Yep.
All right, so Olga, hope you're doing okay. I finished reviewing the paper that you're not supposed to know I reviewed. So anyway, it's sent back off, and hopefully you'll hear back from them soon.
Yeah, thanks. I met an editor today and she told me that she got a review. So we are waiting for another review and I will see both next week, I hope.
Great. All right, take it
Thanks.
easy. We'll see you in a couple weeks.
See
Bye.
you.
So did Luma left? Dang it. Oh, well.
What-
See you
what-
later. Bye-bye.